for example, in touch and all sorts of environmental cues, right, that it then processes with its nervous system. All right, so this was just a frozen view. Let's look how they look in real life here. All right, what you see here is a plate full of worms at all different stages. So a happy family with an adult worm, checking out what's going on here. Let's see how that looks. And all these little things here, these are eggs. These are eggs that have been laced with a vulva that you saw in the previous movie here. And these are all little babies. They've just hatched. They're called larvae, right? The babies and teenagers, and then eventually you get become an adult. All right, so let's look at much more detail. So here's something that will immediately make you aware of the fact that we can do things with worms or flies that we cannot do with humans. And this is this. We can look at embryos in a dish and look exactly how those embryos develop, right? And obviously, I don't need to detail that, I'm sure. We can do that with humans, right, like this. We have a very gross idea only, gross in the sense of a very rough idea, in terms of how exactly the human embryo develops. Here we can study that in the greatest detail we want, and we can manipulate that in any way we want, right? So we can ask, for example, if you look for this, actually let's play the movie first. This is essentially just a fertilized egg, nothing more than that. At this point, it's starting to divide. There are two nuclei that you see here that are marked with these circles, and you can follow life. This is slightly sped up about tenfold or something like this, the movie. You can see how those individual cells divide. They're not really anything yet, but eventually, once they continue to divide, they will make this decision that I mentioned earlier as to whether to become a neuron or a gut cell or a heart cell or anything else, right? Now the cool thing is, as I said, you can manipulate that. You can manipulate that in a way that you could not manipulate more complicated organisms. And you can, for example, take sort of what I'm doing now. You can take a high-power laser, which is literally something we do in the lab, and point that to the cells and kill individual cells and ask what consequence does that have on the development of the whole organism. Are there, for example, cells that provide signals to other cells to allow them to develop appropriately? So those manipulations are all things we can do with these organisms and not as well with other organisms. So actually I'm slightly out of order here. I wanted to show you another movie first that illustrates all the different organ types here. So if you're not grossed out enough yet, you'll have to endure that as well here. You see a worm crawling along. And by the way, let's start this. Look at how it is sniffing around. Oops, let's see. You see how the nose is constantly wiggling around and trying to figure out, where's a good life here? Where can I find something to eat? Right, and you can see here, let's play that again in the end. Here's the gut, here are all the eggs, inside here. This is where it poops from. So we have all the stuff that we also have as well. Right? So all the basic organs are there, right? Now, as I told you, the worm can really sense the environment very closely and do things in response to its environment. And here's one of the ways it responds to the environment. It's very simple. It's very intuitive what they do. Oops. All right. Looks like I promised you something that I may not be able to show you. All right, let's try this. If that doesn't work, then no movie. Too bad. This basically just would have shown you that if you place an attractant, meaning something that they like, and believe it or not, I'm not making that up, they like popcorn smell. They love it, right? And in fact, there's a section in my lab that smells really like somebody's making popcorn all the time. It's a specific chemical that you can find in popcorn called diacetyl that those worms really like for whatever reason. And when you spot a little drop of diacetyl on one side of the plate, worms will immediately crawl to it. They usually do this sort of stuff because they think that something like diacetyl or many other chemicals are an indicator of their possibly being food, right? Food for them means basically, and this is again a little gross, but that's the way it is, they like to eat bacteria. In fact, what we feed them with is a bacteria from our gut, E. coli, right? Everyone
I'm sure has heard about E. coli. They are used for pretty much anything. In our case, they're simply used as food for C. elegans. Now, the fact that you can visualize such a behavior, meaning visualize the ability of a worm to go somewhere, right, and expecting food, now allows you to again manipulate the system and ask what are the neurons that are required for that behavior. And you can also ask questions such as, what are the genes that neurons express that allow them to respond to those chemicals? And again, you can manipulate those animals in many different ways, asking what are the normal mechanisms that make the animal work, so to speak. All right, so now I'm going to show you a more high-resolution view in terms of how you can look now, zoom in even more into worms, and this is not going to be gross anymore. That's actually rather beautiful, I find, at least. What you see here now is, the worm is not as flat as this image suggests. It's just sort of an optical artifact. But what that is we've visualized here a single nerve cell in the brain of C. elegans. A single cell. That's not possible in pretty much any other more complicated organism, just a single cell. You can see here the cell body with the nucleus, where the DNA is. And you can see those processes here, those nerve processes. This is called the dendrite. This dendrite extends here to the tip of the nose. It's actually a chemosensory neuron that detects, chemosensory cues in the environment. This information that is sensed out here is transmitted along the dendrite, transmitted through the cell body into this thing that loops around in this funny way, which is called an axon. And this axon seeks out other neurons with which it makes connections. Okay? So by sort of being able to visualize these individual neurons, we can now more specifically ask, how do those neurons develop? And before I tell you how we do that, I'll show you another neuron type, and that brings us now finally back to Parkinson's.